I will now hand it over to the SLAS Scientific Director, Marshall Brennan, to get us going.
Hello, everybody, and I hope you're having a great day. Welcome to the Biomolecular Condensates webinar, focusing on a novel opportunity for drug discovery with Marta Frigule from Viewpoint Therapeutics. As Liz said, I'm your host, SLAS Scientific Director, Marshall Brennan. We're really excited to have Marta with us to share her real depth and experience in condensate RNA-based drug discovery. And so without further ado, I'll pass it over to you, Marta, to dig into the science.
Thank you, Marshall. So I'll share my screen. Fantastic. So I would like to talk to you today about how biomolecular condensates are an opportunity for drug discovery. So we will start talking about what are biomolecular condensates and how we identify and characterize them, before we actually dig into how aberrant condensates can drive many diseases, and how modulating these aberrant condensates with the small molecule drugs called CMODs can be a next generation of drugs. Finally, I will present an in-house screening campaign approach to find hits to target aberrant TP43 condensates in ALS as a case study. So biomolecular condensates are cellular compartments that lack a physical membrane around them, and they can organize the different cellular processes in time and space. As we can see from this diagram, some condensates are ubiquitous in brown, some are cell type specific in green, and some are condition dependent in red. So these are some real images of these condensates to start having a flavor on how they look like. So for example, these are the centrosome, stress granules. We can see here the nucleoli and nuclear speckles. So biomolecular condensates are mostly formed by a physical process called phase separation. And phase separation is this process by which a solution, a homogeneous solution, then mixes into two phases, a condensed phase and a diluted phase. The condensed phase is enriched for the molecules forming the condensate, whereas the diluted phase is depleted from those molecules. So before we continue, I would really like to emphasize that biomolecular condensates are not protein complexes nor aggregates. So protein complexes have a very specific assembly mechanism and a fixed number of binding sites and stoichiometry, and their size is in the nanoscale. Whereas condensates formed by the process called phase separation, as we have just seen, they have a random number of binding sites and a very flexible stoichiometry. Moreover, their size is in the mesoscale. One key difference that differentiates condensates from aggregates is that condensates are reversible in nature and aggregates are not. Another difference is that aggregates are solid, and whereas condensates can adopt several material states. So now I would like to highlight different features and properties of biomolecular condensates that we can leverage for drug discovery. So first, biomolecular condensates are able to compartmentalize and concentrate molecules, enabling key biochemical processes such as the ones that we can see in this diagram. Activation or inactivation of chemical reactions, sensing, localization, filtration, etc. This extra layer of regulation of the biochemical processes offers a unique opportunity to identify small molecules with new modes of action. Next, multivalency is a key feature of the biomolecules forming condensates. And this multivalency can arise from several different interaction motifs, such as, for example, intrinsically disordered proteins binding to intrinsically disordered proteins, RNA binding to RNA binding domains, but also from multiglobular protein domains binding to multivalent ligands. So because many of these multivalent condensate forming molecules are highly represented in diseases, but they are traditionally considered undruggable because some of them are not amenable to a structure-based drug discovery techniques, such as intrinsically disordered proteins. So condensates offer us the opportunity to drug this such undruggable class of targets. Another property of condensates is that they are able to concentrate a specific proteins and nucleic acids. As we can see that upon phase separation, this condensate forms that in which these purple biomolecules are enriched, whereas the yellow biomolecules, for example, are depleted, right? So this selective environment that condensates offer can be exploited for drug delivery and also selectivity. Moreover, this process is very sensitive to stimuli. So condensates can form and dissolve upon a myriad of stimuli, such as post-translational modifications, changes in the protein synthesis and degradation rates, and environmental factors such as pH, temperature, etc. So all of these stimuli are intrinsic, but as we will see later, they are also sensitive to extrinsic stimuli, such as small molecule drugs. Finally, I would like to point out that condensates are composed of scaffolds and clients. And scaffolds are those molecules that form the foundation of the condensate. Whereas clients, they do not condensate on their own, but they can partition into condensates. And in fact, we can measure how much a client is enriched in a condensate by measuring their partition coefficient. So targeting scaffolds, clients, and also their relative stoichiometries are points for intervention with the potential different outcomes. So we have seen so far that condensates are communities of biomolecules. So two key questions arise. How do we identify them? And how do we characterize them? We can identify them by shape and by size. Regarding shape, they are mostly spherical, and we can clearly see it from these images. However, if they are physically constrained, they can also adopt other shapes. This is the case of the tease granule. We can clearly see that it is not spherical because it is constrained due to the ER. Regarding the size, these condensates, the cellular condensates can range from hundreds of nanometers to micrometers. And here I'm showing you two examples at both ends of the spectrum. So P bodies in this a small scale, and then nucleoli, which are much larger. So because condensates are so heterogeneous in shape and size, characterizing them becomes fundamental. We can characterize them by determining their biophysical properties and also their composition. Regarding physical properties, we can test their reversibility upon stimuli. So for example, in this case, we can see these condensates that they dissolve when the cell undergoes mitosis, and then they form again in the daughter cells. We can also determine their material properties. And if they are liquid-like, they should show properties such as fast recovery after photobleaching, fusion events, and also dripping. Regarding their composition, we can determine the composition of biomolecular condensate using proteomics methods, such as APEX and cross-linking MS, and also imaging, such as using multiplex immunofluorescence. And at Dew Point, we have developed a platform able to identify and characterize biomolecular condensates. So condensates are implicated in many, if not most, of the cellular functions. And this is why aberrant condensates are also able to drive disease. And there are many mechanisms by which a condensate can become aberrant. So for example, let's say that we have a condensate forming in a space, in a time and space that we should not be expecting it to form. Another mechanism would be by ectopic formation. So let's say that the condensate forms in the cytoplasm, whereas in the non-disease state should be forming in the nucleus. Another mechanism is having altered material properties. So let's say that a condensate in the wild type case should be liquid, but upon different, for example, mutations, these material properties can transition into gel-like or solid-like materials. Finally, another possible mechanism is altered composition. So let's say that in the healthy case, we have condensates in which this client red molecule is depleted from these condensates. However, in the disease case, we have this biomolecule within the condensate. So by all of these mechanisms, aberrant condensates can drive several pathological functions, such as the ones depicted here. So they can sequester proteins needed as elsewhere in the cell. They can exclude or include proteins interrupting critical processes within the condensate. And they can have altered composition and or physical properties leading to a toxic gain of function. So because there are different mechanisms that convert condensates into aberrant condensates, we can also think of different approaches that we can use to target aberrant condensates with a small molecule condensate modulating drugs, or CMODs. So one possible approach would be to dissolve or form a condensate. So in this case, we would want the CMOD to promote the formation or the dissolution of a certain condensate. Another mechanism would be modulating their composition. So in this case, we would want the CMOD to include or exclude certain components from a condensate. Another mechanism could be to stabilize a condensate using a small molecule. And finally, we could also think of condensates, of exploiting this selective environment that condensates offer for drug delivery and drug partitioning. So now I would really like to give you a real example on how we apply one of these therapeutic approaches to treat a disease. In this case, amyotrophic lateral sclerosis, or ALS, and how we have designed a screening campaign to identify CMODs in this space. So as all programs in Dew Point, ALS is a high unmet medical need. There is a clear condensate link, and that there is a clear condensate therapeutic approach that we can apply to it. So in ALS, ectopic formation of TDP43 condensates in the cytoplasm drives the ALS pathology. So our therapeutic approach is to correct these TDP-43 cytoplasmic condensates. So overall, we have designed a high content screening approach that looks like this. So we can see how in our screening cell line, TDP-43 is not forming cytoplasmic condensates. Upon addition of stress, we can reproduce the disease phenotype, which is the presence of these cytoplasmic TDP-43 condensates. And then we have screened a library of small molecules in order to find molecules that reduce the number of cytoplasmic TDP-43 condensates. And if we plot the reduction of cytoplasmic condensates per concentration of one of these CMODs, we can see that these CMODs yield typical dose response curve from which we can calculate EC50 values. So let's go a little bit deeper into the assay. So before we actually run the high throughput screening, we did a robust assay validation. And this assay validation procedure is comparable to other non-condensate high content screenings. So during this process, we optimized assay conditions such as cell density, treatment kinetics, readout parameters, etc. We also identified tool compounds to be used in the validation process, and we defined the acceptance criteria. These acceptance criteria allows us to set quality control criteria that is locked in the SOP of the assay that allow us to have a robust and reproducible assay. One of these criteria is the Z prime. We can see that for this assay, we have a good Z prime calculated from the negative controls of the MSO versus the positive control treated cells using a reference compound, and we can see that we have a good screening window. In terms of workflow, so we seeded the cells in 1536 well plates or 384 well plates, depending if we were doing a single point or dose response. Then we dispensed the compound, treated the cells with this compound before we induced the stress for a certain period of time. At this point, we fixed the cells and we image using the high content Opera Phoenix imaging. But this was because our cells contain TDP-43 tagged with GFP. However, if not, prior to the imaging, it's a moment to permeabilize the cells and immunostain them before imaging, as I said. So once we have generated all of these images, we can extract different features using thresholding and segmentation-based approaches. This is a hypothesis-based approach. So, for example, in our case, we would be looking at small molecules that reduce the number or size of cytoplasmic TDP-43 condensates. These images are analyzed in the Harmony software, from which we can extract the numerical feature from which we can then calculate their EC50 values. However, we also cluster the different images that we get using artificial intelligence-based methods from which we can extract different phenotypes. And I'm showing you here an example of how we use AI to identify and group different phenotypes. So in this t-SNE plot, we have represented every image collected from our primary HTS assay. And we can clearly see that the active compounds cluster all together here, which result in no cytoplasmic condensates. However, AI is able to differentiate different clusters within the rest of the space that might be interesting to for follow-up. For example, we can see that this cluster here, in fact, shows larger condensates. So this would be somehow small molecules that promote the formation of these condensates. Another cluster, for example, this one here, really clusters compounds that are toxic to the cells. So in this way, we can further classify and understand our HTS outcome. So of course, this workflow is integrated in a system of different devices that have been carefully designed. And one of the examples I'm showing here on the slide that contains an echo-acoustic dispenser, a Hamilton liquid handling system, and the Opera Phoenix high-content imager. So now let's step back and have an overview of this high-content screening campaign. So the characteristics of the screening campaign, we have actually screened the internal dew point library of small molecules in 1536 well plates in the primary assay and in 384 well plates in the dose response. And I would like to highlight three major challenges in designing and executing high-content screening, high-content imaging screens for condensates. So first is kinetics. So condensates, as we have seen, are very dynamic. But our primary HTS assay is imaged at one fixed time point. So we really need to be thoughtful and careful on how to account for these different kinetics and dynamics throughout the screening cascade. The other challenge is multiplexing. So biomolecular condensates are a community of biomolecules, but we have a limited number of channels that we can image. And this is why it is very important to select the right markers that we want to base our primary HTS assay on. And finally, as all high-content screening campaigns, the automation bottleneck is the image acquisition time. So if we now dig deeper into the screening cascade, so as I mentioned, we screened the dew point library of compounds through our primary HTS and obtained a hit rate of 3.5%. These hits were then further evaluated in a dose response manner, and the hits with an EC50 lower than 10 micromolar were selected, which gave us a hit rate of 1.5. These hits, when they're assayed in secondary assays and counter screen assays, that allow us to then evaluate the different chemical series. Once we generated several chemical series, we could actually run a heat expansion strategy to feed new structurally related compounds into this screening cascade in a positive feedback loop to really get validated chemical series that can be then tested in iPSC-derived motor neurons. And this is what I'm going to show you next. So we can see in iPSC-derived motor neurons how there are no cytoplasmic TP43 condensates in non-stressed neurons. However, upon addition of stress, we can see the formation of these TP43 positive cytoplasmic condensates. And then validated CMODs are actually able to reduce the number of cytoplasmic condensates. And this effect has been validated across multiple stressors, and the effects have been seen with multiple compound classes. So last but not least, this is a phenotypic a screening assay. So now we really need to determine the CMOD's mode of action and run thorough target deconvolution. And Viewpoint's platform is also tailored to do that. So finally, we have talked about biomolecular condensates, how they are a site of vital cellular function, how aberrant condensates are able to drive many diseases, and finally, how CMODs are novel compounds to treat diseases arising from aberrant condensates. So last but not least, translating condensate biology into much needed medicines is a team effort. So I would like to thank the team, thank ALS for the invitation, and I'm happy to take questions.
Well, thank you so much for that presentation, Marta. That was really excellent. I'd like to remind our listeners that they can submit questions through the Q&A box at the bottom right part of their window. We'll make sure that we put those out to Marta. In the meantime, could you talk a little bit about the AI tool that you used earlier in the talk? Did you guys generate that training set yourself? Is that data that's publicly available? Could you give us a little bit more idea of what it took to make that work?
Yes, definitely. So I presented the case study, and it's a team effort. And obviously, this data was generated by our AI team. So I'll try my best, but I can also get back to you later. So what we really want to exploit is to take as much information from the different phenotypes that we get from our high content screening. And these allow us not only to group and define, for example, these CMODs are toxic, but then also to relate certain phenotypes with certain chemical footprinting. So, for example, we could say that compounds that generate this cluster are related structurally in this way. So we believe that this is a very powerful tool to really get the most of the data that you have. And in terms of methodology, so we use publicly available methods as well as in-house developed methods, such as these like weakly supervised embeddings. But yeah, that's more or less it.
Thank you for that. We're actually getting a bunch of questions in the audience now. The first one from Joseph Klim is, in your IPS cell-derived motor neurons, did you see any changes in expression of TDP-43 regulated transcripts that were restored by the CMODs?
In the transcripts?
Yeah, the TDP-43 regulated transcripts.
Yeah, that's actually a very good question. And yes, we did this experiment, but I cannot disclose this information right now. But yeah, this is definitely a very good question. So now this is part of evaluating the function. So now we can see phenotypically that these CMODs are able to affect the condensates in different ways. And now the question is, how do these translate to recovery of the function? And this is exactly what we're doing now.
Thank you. From Mark Norman, where did the reference compound come that showed a positive response in your HTS?
That's also an excellent question. So we ran a non-reference pilot screening first with a small set of compounds that allow us to identify these reference tool compounds to then use and validate the larger HCS assay. However, in cases, so there might be cases that in which we cannot identify this, and then we need to think of other strategies, right? But in this case, we could.
Awesome. From David Lieberman, how was the hit rate of 3.5% chosen?
Here, in terms of cutoff?
That's what I think he's referring to.
Yeah, so this is the number of the compounds that showed a reduction of cytoplasmic condensates to 1 or 2 per cell. So 0, 1, or 2 per cell.
Excellent. From Julio Martin, what kind of counter screens and MOA, I believe that's mechanism of action studies, do you envisage in order to assess the specificity of the potential CMODs and rule out compounds which do not act through any molecular recognition?
That's a very good question, and this is something that we're putting a lot of effort on in really doing a good job in target deconvolution. And we might find some cases in which CMODs act through a specific target, and might be the case that because biomolecular condensates are a community of biomolecules with specific physicochemical properties, maybe we're actually disrupting other processes that cannot be linked to one specific target. And in this case, we really need to use all the tools necessary, secondary assays, all the counter screens, cytotoxicity, really to understand that how specific our compound is to a mode of action, but not to a target. But this, I think, it's going to be one of the challenges, definitely for CMODs going forward.
Yeah, for real. Another question from Albert Lim, are the cytoplasmic TD43 proteins inside the RNA granules?
That's a very good question as well. So there is a little controversy still where these TP43 condensates co-partition with RNA, with stress granules, form different granules on their own. So it seems that, in fact, RNA could prevent the formation of these granules, and that is a TP43 lacking RNA that is, let's say, more sticky and can form these aberrant condensates.
Excellent. Another question from Mark Bronstrup. What is your strategy to deconvolute the molecular mechanism of action of your hits? And that might be related to the previous question too.
Yeah, so let me comment to the best of my understanding and also to the best of what I can say. So we will use techniques such as CRISPR screenings, knockouts. We've thinking of like different different techniques in this sense that do not variate so much from a conventional target deconvolution that you would run in another phenotypic screen.
All right, another question from Mark Norman. What was the stress condition that was used in the high throughput screen?
Yes, and unfortunately, I cannot disclose this information right now.
Okay. All right, and then we had a similar question from Samadrita that I assume that you still can't answer. And then I believe that's everything we have for questions in the queue so far. Does anybody have any last questions that they would like? Oh, sorry, there is one more here from just there. TDP43 has also been observed to form condensates in the nucleus. Do the CMODs affect those condensates as well as the ones in the cytoplasm?
So right now we're focused on the cytoplasmic condensates, and we have compounds that dissolve the cytoplasmic condensates. And we have counter screens to be able to look at all of these, right? So definitely.
All right. Well, if there's any last questions, go ahead and throw them in the chat now. And I'm going to take a second to remind everyone on the call that we actually have a recording of this webinar that will be available on Applied this afternoon. And we will also have a live event, the Careers in Business and Technology Showcase on June 16th. It's free to attend, and the presentations from the SLA community are going to be shared about being competitive, innovative, and creative in pitching brilliant ideas, what we can do to bring a new product to market, and in what ways we can effectively translate great research into a successful startup. So if you're interested in those subjects, please do tune in. And I am not seeing any additional questions, though you're getting an enormous amount of kudos from people listening. And I do have to agree, that was a fantastic talk. I am really, really glad that we got to talk about this. And we will post the contact information for Marta in our channel as well. And so everyone, please take a second to join me in thanking Marta for this great talk, and we'll talk to you again soon.
Thank you very much for the invitation. And as Marshall said, please reach out if you have any other questions. And thank you very much.
Thank you, everybody. Till next time.